Hello guys, today we'll be discussing on the topic electron carriers and electron transport chain in this video. So first we'll discuss on the topic electron carriers and its functions. So by the term itself we can understand these help in carrying electrons from one molecule to the another. Okay, so if various molecules have the ability to accept and donate electrons, so those electrons will be carried out by the electron carriers. Okay, they are also called as coenzyme. They are involved in the cellular respiration as well as ATP generation. There are four types of main carriers, that is main electron carriers. So first is NAD+. So it acts as a coenzyme in the electron transport chain during cellular respiration, where it accepts two electrons and H+ ions to become NADH+. Okay, so in first complex this NADH+ will be converted to or will be oxidized to NAD+, and it donates two electrons to the complex 1. Okay, so in such way this electrons is being carried out by the electron carriers. So next electron carrier is FAD. So it is an important coenzyme which serves as an electron carrier. Same like NAD+, this FAD can accept two electrons and two H+ ions to form NADH2. Okay, so this FADH2 transfers electron through the electron transport chain. So next carrier is cytochrome. So these are the proteins which are rich in iron atom. Okay, so this helps in accepting and donating the electrons. So this is basically present inside the cell membrane, which helps in the electron transport chain. So next is quinones. So these are the lipid soluble molecule which accepts and donates from the one molecule to the another and functions as the mobile carrier. Basically this is embedded inside the membrane, cell membrane. Okay, so it can mobilize from one to another by carrying the electrons. Okay, so the basic outline of the electron transport chain is nothing but here I have taken the example of glucose, energy rich molecule glucose. So when it gets oxidized to the metabolites, there are certain reactions taking place, I mean cellular respiration taking place. So by the each step electrons is being carried out. Okay, electrons is being carried out using the electron carriers, and during that process H+ ions is also being generated. Okay, so those H+ ions will obviously perform in the ATP synthesis, okay, which is needed for the organism for their survival. Okay, here electrons move through the transport chain. They create a flow of protein, generating the proton gradient, and proton is used in the ATP synthesis, as I said. Okay, so the next is electron transport chain. So basically what is electron transport chain in sense? These electrons is carried out from one complex to the another complex. Therefore it forms the H+ ions. So those H+ ions will be given to the ATP synthase. So their ATP synthesis will occur. Okay, by the metabolism, catabolism and anabolism, by the process we'll get the ATP synthesis. Okay, so electron transport chain is carried out in the series of protein embedded in the mitochondria. So it basically contains four complex, that is complex 1, complex 2, complex 3 and complex 4. Okay, in complex 1 it has NADH with FMN, and in complex 2 it has FADH, and complex 3 cytochrome BC is there, and in complex 4 cytochrome oxidase is present. Okay, this electron comes from the electron carrier. So these electrons which is donated by one molecule comes carried out through the carrier and travels through the membrane. So this is the cell membrane. It carries through the membrane and finally it produces O2. O2 is produced in the last stage. So by the production of oxygen, it helps in the production of water Okay, so water will be, this is the last stage of the electron transport chain. Okay, so in first complex, this is the complex one. In first complex, that is NADH, what happens means, there is an enzyme called NADH dehydrogenase. So those enzyme will help in the oxidation of NADH to the NAD compound. Okay, so by this oxidation, it donates 2 electrons to the complex one. Okay, and there is another molecule called FMN. So this will also donate 2 electrons. So totally 4 electrons has been generated with the help of complex 1. Okay, so it is carried out through the electron carrier called as quinone. So this small particle is called as quinone. So 4 protons which is generated from the complex 1 is carried through the quinone, and this is the complex 2, that is where FAD is converted to FADH2. So here basically no electrons or protons has been generated. Okay, so this is it lacks proton pump activity. It lacks proton pump activity, but it converts succinate to fumarate. That in Krebs cycle, it is derived from the Krebs cycle. This succinate to fumarate is derived from the Krebs cycle, which is the second entry point of the ETC. Okay, so mainly you have to understand that no electrons is being donated or accepted in the second complex. Only it converts succinate to fumarate. Okay, so energy less, production of energy is less compared to the other complexes. Okay, so first complex produce 4 electrons, and that is carried out through the quinone to the third complex. So third complex is cytochrome BC. This is cytochrome BC. So it is basically a large protein complex which helps in the proton pump. Okay, it pumps the proton. So in fourth complex also, I mean third complex, it also donates 4 protons. Okay, so from complex 1 it donates 4 proton, and in complex 2 it no such protons has been generated, and in complex 3 it donates 4 protons. So totally 8 protons is produced at, I mean donated at. So these will be carried by the cytochrome. So all these proton will be carried out through the mobile protein called cytochrome C to the complex 4. So in complex 4, as I said already, it produces O2. Okay, therefore helps in the production of water. So in last complex, by the production of O2, it produces 2 protons. Okay, so totally 10 protons has been generated through the electron transport chain. So this is the ATP synthesis. This is also called as phosphorylation, where production of ATP will be generated in the last ATP synthase enzyme. Okay, so this is all about the ETC. Okay, so now let's discuss about the phosphorylation. This is also called as chemostatic theory, which was introduced by Peter Michel. Okay, so according to this theory, electron transfer through the electron carrier through the mitochondrial membrane by the redox reaction and release energy called PMF. So according to this theory, the electrons which are generated by the electron transport chain through the redox reaction will create certain energy. Okay, so those energy is responsible for this ATP synthesis. Okay, so this creates an electrochemical gradient. So these energy will create an electrochemical gradient across the inner membrane. Okay, inside the inner membrane. So as I said here, inside the inner membrane, when the electrons has been carried out, it creates certain energy inside the membrane. So those energy is called as PMF, proton motive force. Okay, this proton motive force is required by the enzyme. Okay, these are required by the enzyme which is called as ATP synthase to catalyze the ATP. Okay, for generating ATP, it needs this proton motive force. Okay, only then it can produce ATP inside the cell membrane. Okay, so this PMF will convert to the ATP. Okay, so with the help of the enzyme called ATP synthase, PMF will produce ATP with the help of the enzyme known as ATP synthase. So ATP is produced by the ADP, adenosine diphosphate, plus inorganic phosphate. Okay, so ATP is generated across the cell membrane through the ATP synthase and re-enter. Okay, so here we can understand when it donates the electron, it produces H plus ions outside the cell membrane. Okay, when it flows inside or when it comes inside, so this part is this part I've drawn here. So that is the last part, that is ATP synthesis. So when this H plus ions are being when it comes inside this ATP synthesis, it produces certain force. Okay, certain force. So mechanical force is being converted to the chemical force, and hence this will create an ATP. Understood? So this proton moves down against the concentration gradient. As I say, this H plus ions is present outside the cell membrane. When it moves across the concentration gradient, so ATP synthesis will be occur. Okay, so it releases certain amount of energy. So FO, this is the first part is called as FO, which helps in the rotation where the mechanical energy is converted to the chemical energy, and ATP synthesis will occur in the F1 region. Okay, so basically ATP synthesis complex is composed of two regions, that is F0 and F1. Okay, so F0 is protein channel flow embedded in the membrane. So in membrane it is being embedded. Okay, so these are the proteins which are embedded in the cell membrane, which helps to rotate the proton. Okay, when it comes inside, these protein will rotate faster. Like how much concentration it is coming inside means, so that force it rotates, this protein will start rotating. Okay, so the express concentration will be higher in the intermembrane space. So in the intermembrane space, this H plus ions concentration will be more, and it creates a certain energy that is called as electron chemical proton gradient. Okay, so F1 is a region where it protrudes the space and the matrix. It is produced outside the cell membrane where it catalyzes ATP synthesis. So here only this ADP is converted to the ADP plus inorganic phosphate is converted to the ATP. Okay, so this is attached to the F1 region. Okay, so these both are stationary phase. Okay, this both are stationary phase. Only it produces how much H plus ions is coming inside means, so in that ratio it starts rotating. So it converts the mechanical energy to the chemical energy. So ATP synthesis will occur. Hope you understood this concept. Thank you for your watching.